So what is the problem? What is the solution? You all can hear from her mouth, rather than I am explaining, because she spent her energy, time, and passion. So she will be able to explain better than how she was able to identify the social problem. How her science and technology background helped her in developing a solution. So once developing a solution is not the end of the story. It has to be implemented, practiced. It has to be taken to the industry, which can bring the product to the market, to the society at large. So that is the story she is going to tell to us in all her ways, revolutions, problems, challenges. Developing a technology in academic environment is not that easy. So many challenges. So in spite of all those challenges, she has come out with the technology. In fact, it was the first technology which was commercialized, which was licensed in industry. The university has got 97 years of history, legacy, but so far the university has not transferred any technology to any industry. Let us accept that. But she has got the credit, for developing a technology which is socially relevant, and which has been also transferred to startup company in Childhood. She is developing that product for the optimizing the miniaturizing, leaving the backbone of the technology, keeping it intact, but they are trying to miniaturize, bring the required finish, that appeal, that all those things, industry requires or customers requires. That's what they are doing. Probably in a year or so, the product may be available in the market. So that is the story. But to introduce some of the youngsters, I would like to read out few lines from her CV. That Aparanji Apsarala Kofarla received her PhD in the year 2005 from Andhra University. We are working on immunosuppressive compounds from herbal formulations collected at Paderu. We have just with so many fauna and flora in this region, and she has picked up some of the medicinal plants and extracted that active ingredient and developed few products based on that research. And currently, she is working as assistant professor in the Department of Biochemistry, Andhra University, teaching immunology and molecular biology to postgraduate students. She received DSP Human Scientist, CSA Food Scientist, and DSP Young Scientist, GP Talwar Gold Medal, FIMSA International Award, first prize in the novel IP Insights Innovative Challenges 2018, anti compound and its making process. To her credit, she is an associate fellow from Andhra Academy of Sciences, Amaravati, and executive member of the Indian Immunology Society, very reputed and very popular society doing great service in New Delhi. She published her work in 21 reputed national and international journals and presented talks in 60 national and international meetings and conferences. She was a recipient of travel awards from CSIR, ESP, Immunology Foundation, Britain, Autoimmunity Congress, Singapore, and FOCAS, USA. She organized national seminars, CMEs, and World Immunology Days. Her goal is to discover drugs from marine natural products for a specific mechanism based on best treatments of autoimmune diseases with minimum side effects. She has contributed research in purification of compounds from marine sponges and soft corals collected from Andamans. She has done all the way, and she was a student and as a faculty here and collected all these samples and conducted research and came up with very innovative solutions. Any addition of inflammatory cytokine levels and experimentally induced arthritis and malaria and potential marine compounds. Development of biosimilars, particularly PH17 cytokines in psoriasis patients. One of your patented technology, portable biosensor for type 2 diabetes, transferred to industry. You could see the scientists here have the passion, the commitment to develop things, and she will do it in the time to come. And she will reach much greater heights rather than me. And I invite Dr. Aparanji to take over and please share your experiences, madam, with all our participants. Sabah Sabah Maria.
Good evening, one and all. First of all, I would like to thank our Purushottam sir. He's also my mentor, technology transfer and all that. Regarding patents, earlier I didn't have much idea about this patents, technology transfer, licensing, and the startups and all that. When we attend meetings, annual meetings, technology, I know that there is technology transfer is great in developing the patented technologies and innovative technologies plays a very important role and brought a huge transformation as discussed by other resource persons. We came to know that information technology transformed the whole business or revolutionized the whole uh, in, the country economy. So this with my research experience, I came to know that information technology and their use will see this way. So I would like to share one of my before explaining the slides. I'd like to share some of my experience. When I went to being a women scientist at Andhra University during the 2008, I went to uh, MS Swaminathan Research Center, Chennai, situated at Chennai. We visited the labs after completion of the project presentations and all that. We visited the labs. Then we came to know that, believe it or not, when the tribal people are doing the experiments, especially biotechnology experiments. I wanted to see, so these without having any knowledge, scientific knowledge, they are screening the experiments. They are doing it. So that means they gave a training institution. What is that? MS Swaminathan Research Center. They gave a training. They developed a skill in tribal women. They they are doing those experiments. What we are doing in the scientific laboratories, molecular cloning experiments and transformations, gene cloning and microbiological examination, everything they are doing. So I surprised to see, and then we asked other researchers there. So then they told that we are doing, being researchers, we are doing only research designing, designing the research proposals, designing the research plans. So we use our brains with our knowledge, and we use to prepare. That knowledge will be used to only to prepare research proposals. But without having the knowledge, that means then I came to know that so our brain is very important. We have to focus on knowledge and how to culminate, how to coordinate the technologies. So now we are in the era of multidisciplinary. With the multidisciplinary approach, how you can solve the problems, so especially these societal problems. With and there are so many solutions. If you culminated the technologies, different technologies, you definitely you can solve that problem. So here, what I need to say, designing a research proposal by systematic. Designing research proposal is not that easy. You don't want to follow some criteria, some plans, some strategies need to be applied. Good proposal. commercially viable. Most of the research people look for what we are doing, being a scientist, I discussed about my technology, then you accept it. I will impress with my communication technology, what I will do. Okay, you will accept it, then you can take it to market. So that is that may not be the success. This will be there always. So being a scientist, so what is the meaning of experiment? Experience you have to. So we are validating experiment, experiments to the laboratory. Okay. And coming to this kind of topic, identification of social problem. What is the social problem? Very negative consequences we have observed in the society, not human population, to be addressed with your scientific approach. Your scientific approach is very important to make that convert that, to transform that idea into a real problem, define structured problem. If there is a that would be a defined result problem that would be conducted into validation before converting it into a prototype. Okay, ability prototype. So next, you all know that invention is the thinking. So to generate an idea, innovation is doing thing. Okay. So the innovation can be considered as an interactive process. So different disciplines sit at one place and discuss things and create and define the problem, structured problem, and then that has to uh, write a proposal, and then by analyzing the data and write a proposal, and that is subjected for and again reframe and subjected for study. So this is very important area we should focus on before coming to the market. Anything. And why we need to develop the technology. So unlike information technology, that takes very less time. What we heard since yesterday morning. So information technology is very, you can say speedy technology. So today we are listening about some, and we may, but what biological the technologies in the biology like biotechnology, nanotechnology, and genetic engineering techniques takes time because it requires laboratory experiments, scientific to validate the proof of concepts. So why we need to develop a technology? You all know that the betterment of society, improvement on existing technology, global community emergence, competition with existing technology with others, and the main thing is to address the societal problems. The technology, there is a need for technology to address the societal. It's a response to the societal problem. You can think and reverse. It's a response to the societal level. You develop the technology that has to be addressed. So and also academic benefit also. And here there are two things. Startup incubators are there and human resource development. So this is startup incubators are all market oriented research. They will as a morning we have also discuss about the market based research. So strategies which we can employ to improve the quality of the product and marketing the product to make money. But being a researcher in the laboratory or being an academician, so we carry out impact based research. Impact based we need workforce and innovative ideas. Innovative ideas. How innovative ideas or problems are generated. For that, you need to have expertise. You need to have lot of information. You need to have knowledge. Knowledge is the basis to generate ideas. Without knowledge, youngsters, I am telling you. So without knowledge, you should go through the number. You should experience as much as possible a number of experiments and number of use literature. Then only you can develop idea. Idea generation is not a simple thing. So Before that, before review of literature, you need to, you need to have, in order to generate creative idea, you need to, have a best quality observation. Without observation, you cannot generate not even a single idea. So you have observation means observing the surroundings, observing the problems, what are there, what is the requirement, observing the society, that means the marketing. What is it? Here the observation means you are doing the laboratory work, but if you are not observing the society, what they want, that is called a marketing research. So there is a link between the observation and the generation of idea. So you need to observe the things, then only. The best example I can tell you, you might have heard about in the school days, Alexander Fleming, who is the Nobel laureate, okay, for the discovery of penicillin drug. So it's such a wonderful drug, antibiotic, not drug, it's an antibiotic. So because of this simple observation, without having innovation, without having technologies in those in 1940s, we could able to conduct experiments just because of observation. It came out. That antibiotic came out. So one day, what he observed, so his nasal, most of the people might be knowing, and his nasal drops fell on a culture on which microbial growth is there. So that microbial cell walls dissolved because of this nasal drops. There is a reaction, chemical reaction between. There is a reaction between the nasal drops because he is Alexander Fleming is suffering from cold that time. So unnoticedly, we are that incidentally that those nasal drops fall onto this culture plate. So it dissolved the culture, microbial culture. So then that question came, the idea came up, why it happened. So every individual questioned himself why this was happened by doing this experiment. So by doing experiment only, there are so many ideas came up. So then he realized that there is an enzyme called lysozyme which cleans the peptidoglycan layer of the cell wall of the bacteria. So that is responsible for cleaning the bacteria and death of the bacteria. So that's why he discovered penicillin, wonderful antibiotic. So but still we are using that penicillin. Okay, so it took 20 years to come into the market. But nowadays, so repurposing drugs and with the advent of this information and technologies and all other tools, we are making the product very easily. Simply the same bottle and changing the cap, color of the cap, we are marketing. Am I correct or not? I may be wrong because it's not class, you are also my colleagues. Okay? So just like a discussion, I'm not business person, I am junior. So that's why. So the same old bottle and we are changing the color of the depending on the demand of the society. So suppose the society is asking red cap, so we'll keep red cap by doing survey. We will keep red cap and marketing. The same thing, same knowledge. Okay, do you agree with me? Okay, right. And this startup startups always are business people always look for restraint by profitability, and we always see the experimentation and experiencing the truth. Okay, so there are always I agree with there are always uh, integrated approach is very essential in order to make a good startup or a business or an enterprise and academia. These are the qualities. Always should be open-ended, should come up with open-ended ideas, knowledge transfer, information sharing, acceptance testing, hypothesis testing, and creative type of activities must be shared between the academia and industry frequently. Then only will come out with a good proposal, research proposal. You can design a good proposal leading to the good product that is viable to the commercialization. And this is a model for industry academia for developing a technology or a research proposal. From academia side, so academia develop because huge knowledge will be there on a particular area. If there is any societal problem is there, suppose say for example sickle cell anemia is there. That is, we all know that. So, though, as a being a biochemist, I know that there is a replacement of one amino acid, that is valine, is replaced with glutamic acid. Simple replacement will brought the property, complete property of red blood protein will be changed. As a result, there is a sickling of RBC, shape will be changed, and that leading to the sickle cell anemia. So nowadays, past Paderu people, we worked on several samples at several people, patients from Paderu area, tribal areas of sickle cell patients came to KGHOP, and that samples were sent to me, and we analyzed the sample using. So because sickle cell anemia could be observed only through microscope, possible only through microscope, but we didn't have expertise in observing the. So nowadays, expertise in the old technology is gone up. Okay, observing the microscopically. That's why we borrowing the new technology. Like in our lab, we isolated the DNA from the blood, and we developed the primers, and using PCR technique, we identified that is the new technology adapted for analyzing the sickle cell anemia patients. Okay, so we gave under subjected for cellular specific electrophoresis, that is different technique, and we subjected the results for analysis. That is interpretation. Okay, that is how we identify the social problem and here locally, and by setting the parameters, by setting the research proposal, research plan, we concluded the results. That is one example. And here the progress. So what this is the model. You all know that together, together, they have to discuss. Academia industry has to discuss to prepare the and finally and review and future plans and what are the cost and benefits. The academia should think of about the progress for funding. Here, the progress for investment and the new technology developed, that is finalized outcomes, and then agreement should be there. This is how the research proposal has to be prepared. This is through integrated approach. It is possible to generate a good proposal, research plan. So we all know that this Covaxin is inactivated virus vaccine. So this is there always because again reduction of the strain takes place. So even that they use it because it's very highly immunology expertise because it produces a immediate immune response. Long lasting immune response is produced only through Covaxin. That's why they're selected. This is there. And with, this is public-private partnership. They discovered this Covaxin and how the problem to technology. So the story about COVID, the recent example for the problem, it's a social problem. Here this COVID, the coronavirus, COVID disease, the social problem, the journey from virus to vaccine, the technology developed. So here there are different steps. In January 2020, it was the diagnosis of first important case, and then you can see about 2020, development of ELISA, 16 January 2021, vaccination campaigns, and then June 2021, phase 3 has, and finally, emergency use licensing for by 3rd November. That is not earlier it was not there. At least 5 years. What is the difference between the earlier vaccine and this vaccine? So earlier they have a sample size for 15 sample sizes. One of the criteria for any research design. Sample size is more. That's why they have taken per pocket and the samples and they came up with a solution. But because of the emergency, so the benefit by this patient is high, but they just by release that implemented that vaccine, how that using that technology, using bioinformatic tools and other things, computational techniques, they analyze the data sitting at home because we cannot spend more time in the laboratory. So that too, in order to culture the virus, you need to have a BCL3 facility. Only in our country, one or two places have that BCL3 facility. NIE, in fact, National Institute of Virology, Pune, is having that facility. That's why they have used to conduct that experiments. So everyone is sitting at home using this competition to analyze the data and this thing and coming to that helped us, that technology. I agree that the technology has put this sector, healthcare sector, and especially in COVID time for the development of vaccines. And these things you might be knowing, impact of technology, current problems, statements. These are the current problems. Poverty will be addressed by using information technology, especially healthcare. I would like to focus on healthcare, telemedicine, and biotechnology, nanotechnology, immunotechnology, bioinformatics, these are very important areas which are very useful for the development of medical devices. For example, earlier we used to isolate insulin from the pig pancreas. It's very expensive. But now, with the advent of molecular cloning, recombinant DNA technology, means whatever you want, isolate gene and put it in the vector and transform it to the bacteria or yeast and substitute for industrial production. And that is, you can produce the product will get very, there is a huge production of the product. So that using industrial techniques, industrial microbiology, industrial biotechnology, you can production is very high. The, the molecular cloning plays a genetic engineering plays a very, very important role for the production. And nowadays we are using that for the production. That's why insulin cost will be reduced. And similarly, growth hormones and factors, education, climate change, agriculture, precision agriculture technologies such as drones, satellites, imagery and IoT, and transportation, electric vehicles, and manufacturing. And additionally, artificial intelligence and machine learning are being used to improve medical diagnosis, decision making. So basing on this artificial, we can make this. So you have the data that. So you can make this. So last lecture we have heard about the surgeon also depends on AI technologies to make whether I should go or see that is the that kind of assistance we are getting from this such type of information technology for the development of so and this Andhra University is gave me a very good platform for doing the research. In fact, because we have been associated with a number of and then renowned professors and expertise. That's why it's also some tribal one Girisan Girisan Cooperative Corporation, one societal problem brought one societal problem to my lab. So that the tribals are facing some one of the major commodities that tribals are depending is gum career. Do you know that gum career, that Kaderu and other areas? During the past 3 years, due to inflow of gum from African countries, being procured by the market of Indian gum career has fallen drastically, and due to this, the gum being procured by GCC from the tribals of Andhra Pradesh could not be disposed, and thereby this is not be able to pass the revenue to face price of to tribals who are depending on this commodity for the sustenance. So that's a big problem. So then they came to me that problem. Then we set some parameters that certain ability, some physical parameters, that is positive, moisture, point, and to add the value. Here, adding the value addition is very important. As it is, if there's a limited value, can do this. Add the value that the same term can be uh, um, value addition will be there, increase the market. And there are 12 mantras or steps for the research. The first point is that is the research question, and this is the systematic approach. Initial review of literature and distilling the idea question to the towards the research problem. Okay, next to continue the review of literature, formulation of hypothesis, determine the basic research approach, identifying the populations of sample also, design the data collection plan, selecting and developing data collection instruments, and choosing the method of data analysis. Here, the bioinformatics tools or statistical or computation tools plays a very important role for the analysis of data and other medical reports. Implementing the research plan and interpreting the results. So, scientific approach gives the outcome and if you follow the scientific approach systematically, you will get a good outcome that is commercially viable, successful. And what is the statement? A very specific statement which clearly identifies the problem should have key variables as well as give some information about the scope of the study. So here the publication of the problem statement takes place after an initial review and the related literature and discussion process. So distillation is very important. The society problem after distillation is converted to a research problem. So narrow down. And then that has to be tested. Okay, for that by developing or designing the proposal. And then you have research question, foundation of the research process, all begins with the question. And these are the qualities for the finding the question, observation. As I said, observation is very important. Then type of research questions. The different types of research questions will be there. You have to identify, you have to categorize each type of work. As if you have information tools, you have technological tools, and you have the information, you have the data. But your brain does so your brain tells which technology has to fit, which tool has to fit for planning, for understanding this data, use data. That data is coming from the industry, from coming to the research organization or academic institutions. So analysis is very important. How you are applying, on what kind of tool you are applying, that makes a difference. Okay, here you need to categorize the question where so it could be a descriptive survey type or relationship question, best variables, difference question. This is the scientific approach you need to follow. And then problem distillation. As I already told you, it's a statement of problem that is very clear. So after refining, after reframing, after distilling social problem into research problem, that statement of the problem should be very clear, concise, and definitive. Then only you can design a plan, research plan. And what is research problem? So this is the example I can give you. Society problem is different from research problem. Okay. So medical doctor and researcher together should understand the disease and only possible and diagnosis is possible. Do you agree with me? Doctor alone cannot give without clinical data. Clinician, by the lab should give interpretation of the results during diagnosis. So that kind of integrative approach will give you a better research this thing. These are the unsolved research problem which I have fascinated about antibiotic resistance. Antibiotic resistance. You might have heard about multi-drug resistance, antibiotic resistance. Suppose a very simple thing. If I got any infection, I'll take without consulting doctor, I'll take the antibiotic. What will happen? So I may pose. It may my infection may be reduced, and I may pose drug-resistant strain to the society. You keep on using the antibiotics. In India, it happens. In other abroad countries, they don't even prefer the antibiotic prescription without consent of doctor. You don't know that. Okay. So, but here, so optimum dosage is very important. Basing on age and prescribe the doctor without you should not use without doctor prescription you should not use any antibiotic. You are changing the strain of the microorganism. You are pushing that strain into the society. That is also a society problem. So that's how you are posing the problem antibiotic resistance. Strain become resistant. Not strain become resistant. What does it mean? It is not responding to the antibiotic in another person. Modified making the antibiotic modified strain into the society. That is a problem. So strain for TB is a currently the TB process is not applicable. We can manage here sits in the immune cells. Actually, immune cells are meant for resistance. It is sitting in the immune cells compartment. That is the big problem. Okay, and also another thing that is being continuously improved in India. That has to be addressed. Kidney diseases. Being a research, when I had PhD, when I was a PhD student, I this is my desire to work on this area. So, 15 years back, I heard about this is a local issue. That is coming. So still there are few experiments were conducted. These are there. Even India visited Harvard people visited Uganda, Chicago district, and then they could not even find out one reason related to this pathogen CPDs.
I am from that region, still no solution.
No, so there is no solution. Unsolved problem. Being a scientist, being academicians, or maybe investors, they should concentrate it. Because simply the glomerulus, the kidney membrane is children పడిపోతుంది అంటారు కదా అట్లా the proteinuria. There is no, cure for that.
So what
unsolved problem.
some studies says is that this is because of the groundwater contamination. There were earlier studies. So Uddanam area is known for this cashew, cashew growing. So there they use a chemical called endosulfan, as a pesticide. So one hypothesis was that this endosulfan has gone into the groundwater. People drinking the groundwater is causing this kidney disease. Almost every village there are about 100 people minimum this kidney disease people. The government is paying pension and lot of treatment. Now recently, Jagannath Reddy is constructing a huge water reservoir to supply potable water to all this Uddanam area. There are more than 800 villages are there. So all these areas, building a potable water storage tank, pumping the water from somewhere here, and supplying this drinking water to all those people. But this is still an hypothesis, is not the scientific evidence is not there.
Not, experimentally proven. Hypothetically, they're telling that the water might be a contaminant, microbial contamination, or heavy metal contamination, whatever it may be. Next, we in the laboratory, the university department of we conducted experiments and animal models and also humans. So look at the difference between the information technology and this technology, biotechnology or other technology, whatever technology. So we are doing on the laboratory is different, entirely different. Okay, if you touch the computer, need not take with the help of computer, develop a tool, computational tools. No need to take any permissions. Okay, but if you develop any blood, there is some, if there is any intervention, then you need to take ethical clearance. Unless until if you take any ethical clearance, you cannot carry out the experiments. Need to answer. But because we worked on psoriatic patients, we collected the skin. 5 centimeter area of skin was and collected, and then we separated them and we cultured the keratinocytes and we produced the cytokines we estimated. So that is a laborious process. Technology is very, very difficult. We need to take a lot of these things. Malaria, and we also worked on animal models, mouse models, rheumatoid arthritis, we worked on diabetes. Because why? What is the reason for that? The system diseases. Multifactorial diseases. So many factors are involved in the development of disease. That's very important to develop it. You need to understand deeper, you need to have a deeper understanding of the subject related to disease. So simply take for cholesterol will be low. If you take this, that will be reduced. We don't accept that. In there and in the market, suppose if you put food in the oven, okay, what is the difference? So simple change, because we study biochemistry, biomolecules, chemistry of biomolecules. Simple change in the So good. Yes, sir. Change the cooperative will be changed. Suppose you look at the group antigens, ABO, simple difference in the linkages, lactose linkages, difference in the lactose antigens. You have a structure itself, property, change in the hydrogen group, property change. So, but put that the oven structure. I believe that the structure of the food change definitely. The microwave change the structure of proteins. Now the food is in the form of capsule. The technology as the technology is growing, the risk will be more obviously. These are the things that are taking place. So I don't want to discuss these things at the time. You all know that it's a very good literature reservoir. Okay, so reading the whatever everything is operating with the generate artificial. So this scientist, this ChatGPT system, other this about this technology, the recent one for translation research. Smart toilets are key for the precision. I think we all heard about the precision and personalized medicine. Just now we were discussing during lunchtime. Now we are getting personalized marketing, just like personalized medicine. So we cannot sell the same drug to take one antibiotic. That is that habit will not be the same norm. I may have different only say depending on the genetic constitution of the individual. I may respond to that. Okay, that is a difficult concept. New technologies are coming up using personalized medicine. So, you understand. Here, the smart objects are the whole, but such passive monitoring has considerations. And how scientists are using artificial intelligence to talk to humans, decoding. The artificial intelligence are helping researchers decode animal communication and bring it back to humans. This is the technology. Okay? Next, coming to our area, nanotechnology. So, nanotechnology is here. So, we know that mRNA vaccines have started to be clinical settings for our species. We all know that Pfizer vaccine. Now it is controversy. Pfizer vaccine that facing a lot of this thing, export issues. Vaccine because we know that mRNA is not being a biologist. We know that this big vaccines. Okay, it's not successful, but risk benefit ratio. They see the risk benefit ratio of the benefit ratio. This is called the emergency condition for the vaccine. That is the first vaccine, mRNA vaccine. And the expert evaluate the technology risks. Here, this is very important in being an expert. Please look for this. That should not be. So, you invent any technology. So, releasing the market. You should assess that it's an ethical process. Medical, if you are that consciousness, okay, result in catastrophic information, actually often damage is with nanoparticles as a therapeutics. Benefit to restoration, intended medical application, technology, so marketing. But there are some scientific gaps have been identified in very recently in nanotechnology. What are the technologies you are set for experimental purpose? They are insensitive. Then how do we define technology? Suppose if I follow the technology is hidden, I may give some specification, the label of the product, and after validation and all that. So how to verify it thoroughly? That is called validation. as a the product or whatever technology. So what are we using still today for nanotechnology? So very recent article has studied their incense life cycle. We have been using other essays like some hypothesis, oxidative stresses have been following still in the laboratories for nanotechnology. So we have got good, our thinking is good. We are getting this techniques. That is a models. But energy, what are the emerging methods to evaluate toxicity of nanoparticles? What are the new technologies? They are nothing but always by attending meetings and by attending this literature, by following literature, what are the every day they have been changing. You know, follow and catch the technology. Cell-based nanotherapy and stem cell technologies and issue of engineering. There is this subject of transformation biology. We have a pluripotent stem cells. So scientists knows that, researchers know that this cell is transforming into the developed cell. Okay, if you know using technology, particular ASL is there to stem cell pool. Pluripotent stem cell pool. ASL is converting the human cell. So we can, it is very easy for you to understand pathogenesis. Same principle can be applied to the diagnosis of pathogens. Okay, tissue engineering, PSRSA, 3D organ structure models, organ and chip, organ chip. So if you study the, so now we are used to, even still we are doing that, following that nanotechnology, the cancer cell lines, still they are the starter cells. I know that they are working with cell lines. Okay, is it okay? Cell lines. Cell lines is not correct. When we use animal models, so when I went to conference presenting, the animal experiment is the same with the human. So if you would that, my response is different, his response is different. But if you conduct it, suppose it's seen any drug, the animal model, how can you say that this drug is effective to humans? So what is covered that is not true because it's not similar, they're genetically different. So, but we cannot do that of all these but there is also state even shows when animal experiments. We face a lot of problems with animal experiments. 10 years experience working experience with animal models. But in between, what is the correct one? So develop 3D organ structure model to conduct experiment. That environment is very important rather than doing in cell lines. So most of the startups they are doing with the cell lines. That is not correct because the environment, how it responds against that particular drug or antibiotic, then that environment you can make it out only through this 3D. But for the screening purpose, you cannot use human, you cannot use animals. In between, you can develop the 3D structure models. This technology transformation should reach the high precision. Accuracy must be there. Then only, and also for the low that is a viable technology. That is called as viable. So, but this is my habit of when I go through the any literature or any paper, so I actually to see the work. So this since I'm my expert is immunology, this Nobel laureate works follow always repeat and follow Nobel laureate works. What they're doing that knowledge is very important because Nobel Prize taking is not a simple thing. Okay, so there's a lot of things there, art of there. So the different resources, information knowledge is very important. Research problem identification or generating an idea of what I need to do. Knowledge is very important. And the immunotechnology is this idea. So we were able to develop different strategies now. These antibodies and T cells. T cells, we all know that it's engineering. They are developing parts of their T cells through which we can treat cancer patients the more effectively, high precision. So the more expensive technology, they are searching for message. regarding science startups. So government support and analyze 75 impactful startups. First day, I gave the first special distribution among overseas agencies. 25% for healthcare and medical devices. No. So we can get points while executing a project or a device or whatever. It's not very important. And come to my innovation or invention, my first commercial technology that is very proud, in fact, trader. Trader also gave me work on share of something. So, medical products and also encourage me to do this product in the market and have much knowledge in the market and also so one person cannot have all these things. Okay. Portable batteries are product is portable batteries of this. So what is here? So this actually was started for this is actually by for working on acrylic compounds we work for 10 years. Okay. But when COVID during COVID situation, so that is a really bad time for everyone. So one family, relatives or family persons, um, that happens. So that time I was seriously thinking about we should able to do something that is very useful to this diagnosis or early therapeutic issue has to be invented. Then we, along with my student, what active and energetic. I think he's also have some skills, competition skills of mark. So I should acknowledge him. He's the same. Like-mindedness is very important in research. So Archie is also he had to be developing some software tools and all that. So we developed within a period of 18 months. We developed a prototype, laboratory prototype, and validated validation that at ATZ, and we gave place for working on that. And we did it with the help of vice chancellor. So they helped me for technology transfer. Very helpful in that. So hard work. How I mean this actually బిజినెస్ అనగానే నేను ఒక చాలా మంది everyone doing incubation at why don't you go to industry? Why don't you keep industry? Or I thought that here because not also influences. My father school teacher, మా మేనేజ్ చేసినప్పుడు కూడా business under he used to treat as I don't know. Maybe not systematic. It's just like that. I'm pushing them. So again, my decision was not that be complicated. Studying and తర్వాత this is the growth and dignity of the people. It takes so much to the society. Business is not good. So so one thing that I did this idea, even just he is very much interested. He's very good at non-invasive, but you add this thing, that thing. So, but the result will apply properly and we I got like this and I cannot come till at the end of it. Because that impression is somehow it is in my brain. That's why I could not make it. But it dedicated me is the one. That's why I Pushottam is part is important. It's not only the course. Okay, on that we can add just how food preparation, how we add spices. We can add technology and spices, not only the basis core subject. So we can add all these which we can learn about science. But it's the specifications. This technology is specific.
Sure.
But before that, just
Okay.
for a
Okay.
sugar levels. So it's just
ఇప్పుడు పంపించిందా పోయిందా? ఓకే. అలాగే
device will be there.
అలాగే
We thought that my student, we thought that so same device, why don't we apply it for the multiple parameters? Why don't we set this multiple parameters? These will know this 10,000 for cold infection, 10,000 for test. Okay. So any business, so single whatever device, it's a point of not multiple analysis can done. Not the set that is our very natural. This is what analysis structure and other. These are the specifications. The second thing, I worked for 18 months. This I worked for on 10 years on this. We have collected nearly 40 samples, export partial samples with the Association of Andaman Nicobar Island. Because you see, to get what is that? A collection of samples, marine sponges and soft crystals, the climate, Andaman, in restricted areas. So out of 25 proposals, we applied research proposal. Okay, only 5 proposals got approved. So that among those 5 proposals, the one proposal that got accepted, the DSC people gave me 1 lakh for only for collection of the samples. Gave me separate board, divers, and we collected nearly 40 samples. We could able to, but it's a very difficult process, purification and this thing. Now with the technology, and we need to have manpower also. Okay, so to isolate, with the help of MSc students, we were able to do 10 or 11 projects, MSc projects. We have purified some compounds and look at the parameters. We might have a cytokine storm in COVID time. So these cytokines are plays a very, very important role, a central role, number of diseases. So these cytokines are nothing but they are proteins secreted from the immune cells. Not only immune cells, other cells also secreted. By looking at these end products of the cells, you can assess the disease. Earlier we used to, how do you detect the disease? In good olden days, you go for either blood or urine for clinical detection of any disease. Very simple. To observe the patient should be there. But now we are in the era of telemedicine. Okay, symptoms of the medicine chapter. Very simple. Okay, but these are very important. We said why? Because they are so inflammatory test. So inflammation, you see, inflammation is a part of any disease. Without inflammation, there is no disease. Any infection, inflammation is there. Gastric ulcer inflammation there, cancer inflammation there, arthritis inflammation there, cancer inflammation. Thing, cancer is inflammation is common. So first, in COVID also inflammation is there. The inflammatory cytokines. So biosimilar is a prominent area of research. So we worked on that and we estimated using the IPM technology that is multi-VSA, flow cytometry, and we analyzed and interpreted the results. Okay. Also patent file. So this is the last slide. Partnership, improving landscape. Very important. These are the things that we must be knowing. Indian Innovation Growth Program, the DSP is giving, aimed at, they're very specifically giving the funding, aimed at enhancing the Indian innovation ecosystem by enabling innovators and entrepreneurs through different stages of idea generation, innovation, and acceleration to develop technology-based solutions for future. And there is also another fellowship that is Prime Ministers, especially youngsters, Prime Minister's Fellowship Scheme for doctoral research by SERF, BSc, Public-Private Partnership Scheme for doctoral research between Science and Engineering Research Board, and there are, you might be knowing, Scheme for Funding Industry and Industrial Relevant R&I, RRED, the DST, to address industry-specific problems. And today I received this letter from POSIS. POSIS is Federation of Invitation for attending Federation of Clinical Immunology Society. So only 3 to 5 people they'll select every year in one state. Okay. So this year they are organizing. The POSIS is organizing at Boston, USA. So I got invitation today morning from Federation of Clinical Immunology Society for presenting that. Thank you very much. I'm very happy to see you all. Taking class for the students is different, but sharing with our information with our colleagues and is different. Okay, thanks. Class అంటే restricted area మీద మనం టీచ్ చేయాలి అందుకంటే Thank you for your patience.
I love your talk. From slide, but I both the audience out here because I guess I can blend you here anymore. Thank you. Thank you.
Any questions? Yeah, sir.
And what are the conditions that are creating this problems or finding the problems? And what are the conditions that are we have to focus on able to form this picture? That if you study in that fashion, I think we will be able to meet some more focusing to get our
Setting parameters are very important through systematic scientific approach to reach that market. What I mean to tell you. Thank you once again.
Sir, one point I'd like to share with you. Whatever technology she has developed, the sensor for the diagnosing the type of biologies, not early stage. Whatever money was required to develop the technology, the entire money she spent from her pocket. No, university has not given a single fee.